So my name is Silvio Bicciato, and I'm an associate professor of bioengineering at the University of Modena Reggio Emilia, even though I've been mostly working in bioinformatics for the last, say, 15 years. Before I get into the serious stuff, please let me just spend a minute to tell you a little bit about Modena, just because maybe most of you don't really know where Modena is. Modena is a small town in the northern part of Italy. We are located in between, say, Milan and Rome, close. We are something like 20 miles away from Bologna, which is the major town of this area. And let me tell you that although we are not that famous for computational biology or bioinformatics, I guess that we are definitely famous for what we build in Modena, just because both Ferrari and Maserati has been designed and are now assembled and built in Modena. Actually, Ferrari has a factory which is 10 kilometers out of Modena, while Maserati has its own factory in town. So Modena is famous for their cars. I'm not going to talk about cars today. So what I would like to do today is to present MDP, the Mutation and Drug Portal. MDP is basically a web-accessible database that we deploy, and that combines the cell-based NCI-60 pharmacological screening with the genomic data, which have been extracted from the Cancer Cell Line Encyclopedia and from the NCI DTP project itself. Despite a lot of talks about cancer cell lines as models of cancer, and despite the inability of this model to reflect many of the aspects of a drug's behavior in the human body, nevertheless, cancer cell lines have been the most widely used models to explore the molecular basis of drug activity. And since the pioneering work, the pioneering panel of the NCI-60 project, several major screenings of the combined genetic profiling with drug testing have been created to investigate how genomic portraits can shape cancer response to therapy. Basically, all of these cancer cell line screening adopt two major approaches. The first one, which is the one we based MDP, is investigating in parallel the molecular profiles of untreated cells with their response to various compounds, trying to assess or to predict what is the intrinsic cell sensitivity or resistance to various drugs or to various potential drugs or potential compound that could be potential drugs. And this is basically the approach that has been adopted by the NCI-60, by the Cancer Cell Line Encyclopedia, and by the Sanger Institute screening, whose data are contained into the Genomic of Drug Sensitivity in Cancer portal. The other approach, which I guess is well known to you, is the one where cell lines are profiled both before and after the treatment to assess how their molecular profiles respond to the perturbation by the various agents that has been tested. And this is the idea that was first behind connectivity map, and as far as I understood, that is behind the actual links project. The data of all these genetic features and of the drug sensitivity are mostly publicly available online, and there are several different dedicated data portals that contains not only the data but also the tools that can be used to generate the association between the genetic characteristics and the drug treatment. I don't think I need to name any of all the computational resources of the LINKS project. It has been really a pleasure to be at your symposium last 40 days ago, more or less, in Miami. So let me just consider the tools which has been associated to the screening of untreated cell lines. So basically, the first one is CellMiner. CellMiner is the data portal that can be used to analyze and to download the NCI60 data, all the drug activity as well as the genetic or genomic data can be analyzed through this portal. Similar to CellMiner in some sense is Genomics of Drug Sensitivity in Cancer web tool, which contains the data that has been generated during the screening led by the Sanger Institute. And finally, the CCLE site at the Broad Institute contain all the data from the Cancer Cell Line Encyclopedia. So basically, all this data are available, but despite their broad availability, most of the data of these data are still heterogeneous. There has been a major study published a couple of years ago that compare the concordance between the different screening, especially between the Sanger Center screening and the CCLE screening, and in this study they highlighted point of strength, of strong concordance, as well as some weaknesses in terms of heterogeneity and lack of concordance. Most of the lack of concordance sometimes is in the terms of which are the investigated cell lines and the compounds and of course the type and sometimes the quality of the genomic data that are available. So if we consider, for example, NCI60 and the CCLE, why the CCLE contains, say, the most likely complete compendium of cancer cell lines. The CCLE, the NCI60, sorry, provides the largest variety of compounds, and if you consider the number of cancer cell lines which have been profiled from the NCI60, the number is extremely low as compared to the CCLE side, as well as the number of compounds in the Cancer Cell Line Encyclopedia is extremely lower than the number of compounds that have been screened using the NCI60 during the NCI60 effort. At the genomic scale, while I would say both screening provide basically similar gene expression profiles. There are slightly differences between all the two slightly different platforms that has been used, but I would say there is a high concordance of signals and of number of genes, at least considering the common genes between the two different Affymetric platforms. But when we go down to the DNA sequencing information, the DNA sequencing information is extremely different. Because in the case of the CCLE, there is a high quality DNA sequencing data, but which are limited to less than 2,000 genes, while in the case of the NCI60 data, there is a profound sequencing, but at a lower quality. In general, let me change the pointer option. In general, these two different screening, while having several different cell lines in common, several different genomics data in common, don't really are not interconnected between them. So to overcome this source of lack of interconnection between these two resources, we design MDP. So basically MDP is a public data set, is a public database that bridges NCI60, uniquely large number of drug sensitivity data with both CCLE and NCI60 gene sequencing information and microarray gene expression profiles. So basically, during this talk, what I will do is to illustrate what is this Mutations and Drug Portal in terms of its architecture, how we build it, in terms of data content. I will give you some hints on the statistics that we use that we are using to query this data, and some snapshot on the web interface of the portal. Then I will give you a couple of demonstrations on how MDP can be queried to identify drugs which are active in cancer cell lines, which are carrying specific mutation in specific cancer genes, or how MDP can be queried to identify genetic markers which are associated to sensitivity or resistance to a specific compound. Finally, I will discuss, and I hope to discuss with you about possible connection between this type of data, this portal, and the data and the tools of the Links L1K project. So, going to the structure of the portal, as I was saying before, MDP idea is to bridge NCI60 large number of drug sensitivity data. So we are talking about more than 50,000 annotated drugs which has been used on the panel of NCI60 cell lines. So bridge this drug sensitivity data with the genomic information and the sequencing, the microarray and expression profiles that are available from both CCLE screening and the NCI60 screening. In this way, basically, what MDP can do is query the drug sensitivity data for almost 50,000 compounds on the base of both CCLE and NCI60 sequence cell lines. The guiding element that we use to link the pharmacological information to the genomic data is, of course, the set of common cell lines between the NCI60 drug screening and the genomic and transcriptional information that are contained in the genomic repository. Of course, depending on the type of data that we are considering, say CCLE genomic data or NCI60 genomic data or gene expression profiles, the number of cell lines which are in common, so the guiding element can vary from the total number of more than 100 cell lines, which has been used to test these 51,000, almost 51,000 compounds. These pharmacological and genomic data can be queried to identify, for example, drugs which are correlated to gene mutation, and this is the from gene to drug type of analysis, where you start from genes and mutation and retrieve statistically active drugs in terms of cytotoxicity. On the other way around, we can look for gene mutations which are associated to a specific drug response. And this is the from drug to gene analysis, where you start from a drug or from a set of drug and retrieves statistically significant genes and mutation. Lately, in the beta version that we are working in, but that will be deployed in a couple of weeks, you can also use gene expression data to look from signatures which are associated to statistically active drugs. Starting from a drug, once an active drug has been identified, starting from a gene, so from a set of mutations, so coming from a gene to drug analysis, both using CCLE data or NCI genomic data, what is possible to do is to identify differentially expressed genes, characterizing those cell lines that are sensitive to the specific drug as compared to the cell lines which are resistant to that specific drug. So which type of data do we use to construct MDP? Well, MDP has been constructed using basically the drug response file, the GI50 data, which can be derived from the NCI60 DTP portal. This data are coming in terms of log minus the log 10 of the GI50, so the drug activity as 50% the growth inhibitory level. And before using the data, we back transform them to GI50, then take the log 2 and finally normalize this data by subtracting the average of the log 2 GI50 values across all the cell lines, so that to get what is called the relative response sensitivity index of the cell line to a specific treatment. This is for what concerns the drug response data. For what concerns the genomics data, well, genomic data and variant have been downloaded directly from the CCLE website and from CellMiner for what regards the NCI60 data. While gene expression profiles are available in Gene Expression Omnibus, and there you can see the two different series that contain data using the two different Affymetrix platforms. So, based on the drug response data, we label or we characterize each pair of cell lines and drugs into two major categories. The one we define as good response, all those pairs of cell lines and drugs for which the relative sensitivity index is smaller than a given threshold that we set as the two standard deviation of the left tail of the distribution of all the RS values, and as bad response, the pairs of cell lines and drugs otherwise, with an RS larger than twice the standard deviation of the right tail of the RS distribution. Basically, responders and non-responders based on the pharmacological data. On the other side, looking at, considering the genomic data, we classify the cell lines in cases and controls, where cases are the cancer cell line with a specific set mutation, while control are the cancer cell line without the specific set mutation. And here, by set mutation, you can consider set of G mutation, set of SNP IDs, or set of expression profiles given a specific gene signature. So basically, based on this classification, all the different compounds or all the different mutation depends on the type of analysis one is performing, but just suppose that we are looking for drugs which are associated to a specific mutation. So compounds can be ranked using a score, which is defined by the fraction of good responses in cases. So those cell lines with the mutation which indeed respond to the drug, the fraction of the good responses cases, considering the total number of cases, multiplied by the fraction of the bad responses, those cell lines which do not respond and are in controls, meaning that are not carrying the specific mutation. So basically this score can be used to rank the drugs in terms of specific response to a given, in a set of cell lines with a given set of genomic characteristics. Of course, the statistical significance of this ranking can be assessed, can be computed using a one-tail Fisher exact test for testing the enrichment of good responses in cases as compared to bad responses in control, given the total number of bad responses in cases and good responses in control. As to the web interface, MDP is available at mdp.unimore.it. Again, as I was saying before, in a couple of weeks we will deploy a second version which will be slightly more complete. We will not show major differefices in the engine part, in the analysis, but they're mostly few refinements in terms of quality of the graphics and setup of the web pages. But anyway, starting from this website, it's possible to perform queries. Again, in either direction, meaning that we can look. We can start from gene and look for drugs. We can start from a drug or a set of drugs and look for the genes which characterize the cell lines which are sensitive to that drug. Or we can start from gene expression signature that identify drugs. And in all of these three different cases, we can use the genomic data which has been obtained by the CCLE or the NCI-60 screening. So one can choose between less genes, more robust as genomic data, more genes, slightly less robust as a quality of genomic data. In the query page, the user can input multiple gene names at the same time, so multiple SNP IDs, so to explore, for example, the drug sensitivity to combination of multiple genes or multiple single nucleotide polymorphisms. The same can be done for drugs. Both of the input lines are auto-completing, so you just start writing down the name of the drug, and you will have a complete list of drugs that are available for the analysis. And the same can be done using a gene signature, which is composed normally and as in all the other tools by a list of official gene symbols. Once the genomic entity has been selected, the query can be limited to subset of tissues, meaning to subset of cell lines, and to subset of variant classification in cases there are different variants available for that particular gene, for that particular genomic entity. So this is mostly the input web interface. Now I'll go through the output and the result interfaces, showing you a couple of examples. One example is just to make an example that we use to validate the tool looking for known dependencies, while the other one is an example of how we use this tool to identify new association. To see if indeed MDP was able to retrieve correct drug and genomic variance association, we use an example mutation in BRAF, so we look at BRAF somatic mutation, and in particular we focus on the B600E mutation within the kinase domain, which has been frequently found in melanomas and in several other cancers, and for this reason FDA-approved specific BRAF and MEK inhibitors for the treatment of diseases such as melanoma, and this BRAF and MEK inhibitors has been validated in some clinical trials. So one of those has been published in this paper that I showed you here. So basically what we did, we interrogated MDP, searching for drugs with selective cytotoxicity for cancer cell bearing the BRAF mutations. And when considering all the tissues, the analysis basically identified more or less 190 drugs with significant selectivity for the BRAF mutated cell lines. And what is interesting is that among the top 10 score ranked molecules, we identify several BRAF inhibitors as Dasatinib, Vemurafenib, or hypotemycin, which were indeed the drugs reported in the clinical trial published in the New England Journal of Medicine. Here you can also have a screenshot of the web interface in terms of results. Results are returned as a list of molecules and drugs. Here you have a list of different drugs. Here you have a list of drug families that show statistical significance activity in cancer cell lines that are bearing the specific mutation on the selected genes, which are recapped over here. Results can also be shown using graphical and interactive representation of drug annotation in terms of drug family, of the score distribution of the different drug with significant and higher scores, of the statistical significance of the different drug, and also the gene expression level of the heterogeneity genes in the specific tissue, considering cancer cell lines sensitive or resistant, wild-type resistant or mutant and sensitive to the specific drug. So you can also inspect which are the gene expression levels of the specific genes that were used to interrogate the database. So in a similar way, we could validate MDP on a number of other examples, querying for different genes with different variants in different tissues, and recalling the known, at least, pharmacogenomic association. So as I was telling before, MDP can be queried in either direction, meaning that I can also interrogate and I can also input a compound and look for genomic variants that may be related to the sensitivity or resistance of the cancer cell lines to the given compound. Here is the counter example of the previous example, meaning that we query MDP using the BRAF inhibitor Vemurafenib, and MDP return BRAF in the skin as the most significant gene related to the sensitivity to this inhibitor. So once validated on few known association, we use it. We try to use it to identify new association. And an example, we query MDP to identify compounds that might be able to inhibit the proliferation of cancer cell lines which have an aberrant nuclear YAP activation. YAP are transcriptional cofactors that in tumors escape the control of the EPO pathway and normally keep them or control their relative abundance in the cytoplasm as compared to the nucleus, bringing these two guys to proteasomal degradation. And in tumors, yapentase escape the control of the EPO pathway and activate their transcriptional program, which is associated with cancer stem cell properties, metastatic potential, and so on and so forth. So among the members of the EPO pathways, so among the members that are able to control YAPTA's localization and thus quenching their transcriptional activity, mutation in the NF2 gene have the most frequent alteration that can impair YAPTA's control and that can promote YAPTA's transcriptional activity. Mutation in NF2 gene causes NF2 transcriptional inactivation, as you can see here from the gene expression level of NF2 in mutant and wild-type tissues, in all the tissues which are available in these cancer cell line models, and a snapshot on specific tissues, and at the same time NF2 impairment and mutation causes the transcriptional activation of YAP's transcriptional program, as you can see here in the NF2 mutant tissues. So based on these premises, we use MDP to seek for compounds that could specifically target cancer cell lines bearing the NF2 gene mutations, and thus targeting cancer cell lines with an aberrant YAP activity. In particular, we query the association between NF2 missense and nonsense mutations and drugs on all the cancer cell lines using the from gene to drug approach in using CCLE genomic data. Doing this, what resulted was the identification of only two classes of drugs significantly associated to drug sensitivity in cancer cell lines with NF2 mutations, and these two families are statins and the analogs of imatinib. Actually, the result was partially surprising, meaning that just the year before we work on the demonstration that statins are indeed able to induce YAP's nuclear exclusion. As you can see here in a panel of cancer cell lines, the treatment with a specific statin is able to limit the nuclear localization of YAP entats, and this is achieved by statins blocking the upstream, some of the upstream signaling pathways of YAP's activity, which is a piece of the mevalonate pathway. However, what is interesting is that following the MDP indications, we treated the cancer cell lines not only with statin, but with an imatinib analog, and with the combination of the two. And what we saw is that indeed the combination of statin and Dasatinib, we use Dasatinib as an imatinib analog, are able to enhance YAP's nuclear exclusion, as you can see from this plot, and this effect is basically tissue independent. Moreover, the combination of both statin and Dasatinib are able to block YAP's transcriptional activity, as indicated by the lack of expression of several YAP's target upon the administration of these two compounds, and also the combination of statin and Dasatinib is much more active than the single drugs alone in inducing apoptosis. So, to conclude, MDP is a public available resource that combines genomics and the pharmacological data on cancer cell lines that derive from the NCI-60 and the CCLE screening. And it can be used to identify compounds which are active in cancer tissues which bear specific mutation or specific transcriptional profiles. We prove MDP reliability, identify some already clinically proven gene drugs pair, and validating in vitro some of the novel associations that were dig out by MDP. What we are doing now is basically try to explore if we can find some connection between the result which can be obtained using MDP, mostly focusing on mutation, and the expression data of the Links L1K project, and the tools that you develop during this project. In particular, one idea that we were working on is if it's possible to identify drugs for a sort of second-line therapy, meaning if we can identify compounds which are able to revert signature of resistance in cell lines that share the same genetic background in terms of mutation but respond differently to a drug. So in the beta version that we are currently testing, we have this module here where it's possible to identify up and down regulated genes in cell lines which are resistant to a specific drug as compared to their sensitive counterpart, and these sensitive and resistant cell lines have the same genetic background. So they are resistant, but they don't have the mutation. Actually, sorry, they are resistant while having the mutation, while some other cancer cell lines are sensitive while having the same identical genetic background. So basically what we can do is perform some differential expression analysis on these two populations, which differ simply because of their different drug response. And we can identify lists of genes, gene signatures that we are currently able to supply directly to your tools through the APIs of the links tools. So we can directly query, for example, Enricher or the L1K data through the L1K CDS square tool and return compounds that in principle should be able to revert this sort of resistant signature. This could be an idea. We were just wondering if it's feasible, if the tool is actually in place, but we are still testing the type of results that are coming out. So this is basically it. So I thank you all for listening, and I'll be happy to take any question that you have.
So can I ask you a question? I'm curious. So you're looking for first in this last application, if I got it right, for transcriptional signatures of resistance, right?
Yeah. This is just an example.
So
or you can go the other way around, transcriptional signature of sensitivity.
Right. Oh, okay. So which data are you using to construct this signature?
We are using the gene expression profiles of the untreated cell lines, meaning of the genetic background of the cell lines which are sensitive or resistant. So basically, imagine that you have a subset of melanoma cell lines
I'm sorry, I just meant which resource you're using. Is it these are CCLE transcriptional profiles? Or?
Oh, well, actually, the transcriptional profiles are coming both from the CCLE or from the NCI-60. You can use both. You can use them all together, meaning that we have tools that perform some sort of meta-analysis, so we are able to merge different Affymetrix platform. Depends on the number of replicates that you want to have, just because with the CCLE, as far as I remember, you have a smaller number of replicates per each cell line, per each type of tissue, while NCI-60 give you something like from 4 to 6 different replicates per each cell lines, which make easier the analysis of the differential expression. In terms of signal, they're pretty much the same. They are pretty well comparable. In terms of number of genes, when we use the NCI-60 transcriptional data, we lose few genes, but there are not that many to compensate for the lack of replicate that you have with the CCLE. But it doesn't make much of a difference. Again, we can use them all together if we want. We can fuse the two transcriptional data set together as we did before with several different cancer databases. So we have a function in R that allows us to merge all the different U under 33 generation of Affymetrix arrays. But they are all the gene expression profiles of untreated cell lines. So that's the reason why looking then and submit this data and match this data with your data, which are data coming out of after the treatment. So we want to see if there are compounds that switch off or turn on, depending on what we are looking at, these signatures.
Yes, Peter.
Hi.
Yeah, hi. Great, very interesting. Peter Hornbeck here. I was curious, this is it's not so much directed at you in this talk, but it's sort of to the links committee, the links consortium. There are some really great resources coming up that are doing mass spec analysis of protein levels, of course. When I hear a talk like this, I say, oh yes, but I would really like to know the protein levels and the protein responses, for instance. Yeah. I mean, you can have, destruction of proteins that goes very quickly, and lots of changes that can go on that won't be detected by this. And for instance, Aled Edwards and the, what was it called? I guess it's the Structural Genomics Consortium. In Nature Method this year, that looks really terrific in terms of starting to put together these profiles of protein expression based upon mass spec, say, spectral counts. So my question is, are we trying to incorporate that data specifically?
Not immediately, meaning that actually this is a project that started more or less 1 year ago, and that was motivated mostly by the fact that we were looking for compounds able to, we are working mostly on YAP tasks, so we were looking for compounds able to in some way interfere with the YAP activity. Starting from there, we start building this just because we were looking for mutation from drugs. So we start going through these different resources, and right now we ended up with this version of the database, and building on it, we are now looking at different, if it's feasible and if it's interesting to incorporate different types of data, and definitely your suggestion is a great one. But no, in principle, we started with mutations, so we haven't thought about incorporating anything else but mutation. That was in the beginning, and that is actually the type of data that you can query in the current version of MDP. If you connect to the website, you can query for mutations, just because that was the focus. Then we added the gene expression profiles, just because getting in touch with your data, it was sort of mandatory to try to incorporated gene expression profiles, and now your suggestion is definitely a great one, and we need to take a look into it. We are also trying to see if what type of data will be coming out from the NCI, just because there was in last week issue of Nature the news about the newest NCI, sort of NCI 60 screening that will be deployed in late this spring, and so we were looking also at some other type of data, like data from PDXs, and then see if it's possible to incorporate also those data. Yeah, no.
My question was actually not so much directed to you, Sylvia, but it was mainly, I think, to maybe the links. So Mario and others who may know, if we're actively trying to pull in this wonderful resource that information that's becoming available rapidly.
Yeah. Yeah.
So, Mario, still there?
I'm here. Well, okay, so DCIC is looking to integrate all available data that is at our disposal. You know, there are so many things going on within the three groups that I think there are efforts in these directions, but I'm not quite sure if we are doing exactly what, say, Silvio is doing. So, and then the question is obviously what some of the proteomics data that came out of links basically came from you, Peter, and you know best how to maybe integrate this stuff with these mutational profiles and other things. So
what I've gotten really excited about, though, is I heard a talk yesterday about Alan Edwards and Sachdeva Sasitu and Shohei Koide's work, and large numbers now of profiles of protein expression based upon, say, spectral counts. Okay, so that's opposed to PTMs. Okay, so here I would be wanting to look at maybe rapid changes in proteolysis, you know, ubiquitination and those things, and which won't be visible at the gene profile level necessarily. So that was really my question. And I became aware of this just only recently, so I haven't talked to you since I saw it. So I could send you a link to his paper.
That would be great, yeah.
Yeah.
So one thing that comes to my mind, and I'm sorry for maybe changing the direction, we can go back to Peter's direction. When you say, you know, mutation of BRAF or a mutation of a gene, how do you figure out when there is a meaningful mutation in a gene? Is this just aggregation of all possible variants,
yes.
Or do you assess, are they functional? Is there
No, actually, when you see in the portal when you can select the type of variant, is basically it's basically the variant which has been listed for that gene based on the type of genomic data that you selected. And for this reason, the type of variant classification vary when you change the gene you select, and vary when you change from CCLE to NCX60, so you change the type of genomic data. We simply took the variant classification that were labeled by the original data set, but we didn't make any external evaluation of the type of if the variant was functional or not, or we didn't. You can simply select whatever is available, and of course the way to use this is to select what you suppose could be meaningful and functionally relevant variant. We don't really put our hands on say the biological interpretation of the data.
If I may ask one more question, so I guess CCLE also has copy variants, right? Copy number variants. Yeah. So you don't use that.
No.
I mean, is there a philosophical reason, or it's
no? Actually,
later.
No, actually, we didn't use that just because, as I was telling you in the beginning, we were mining both CCLE and NCX60 exome sequences to look for things we care for YAP and TAS. So we started building on those data we didn't consider the copy number data, even though I've been working on copy number data a few years ago for a while. Only lately we had the gene expression profiles. Now I'm reasoning if we really want to keep building on this and integrate more and more data as they become available, or those data that are unavailable, as you said, the copy number, for example. Or if we want to stop here, it's still sort of work in progress to decide where we want to go with this tool. If we want to limit to this, or if we want to keep building it. We are still monitoring if it's interesting, the tool by itself, if people are using it or not. And so we deploy it, we publish the tool last November, October and November. So we are still trying to understand, the type of bioinformatics we are doing is mostly data analysis more than building tool or coding public available packages. So this is a slightly different track of my lab.
Okay, great.
Slightly different from what we used to do. So we don't even have the competences in coding. We would definitely need some computer science guys, which are not that easy to get aboard, and so we're spending a lot of time in making interfaces, which is slightly different from what we used to do.
Thank you, Silvio. That was really good. Thank you.
Thank you. Thank you. Bye-bye.
Bye.